have amazing speakers and a live demo of Dynamica. So you're joining today a practical guide to imaging in radiopharmaceutical trials. Obviously, this is one of the hottest areas of therapeutical development right now, and also extremely complex technically and in many other aspects of it. So we thought it would be very helpful to bring a team of experts to really speak about it. And today we have Eric Ray, who's the Chief Scientific Officer of Rapid Dosimetry, and Jaron Blonko, who is Chief Operating Officer of Cellectar, joining me, and Shreya Nair, who is a product manager at Image Analysis Group. Excellent. We have more people joining in, so as they come in, we will just accept everyone. So we have the very first guest speaker to start it off, but I first give introduction about Image Analysis Group. So we are a specialist imaging CRO, partnering with biotech and pharma companies to support clinical trials in imaging across multiple therapeutic areas, which would include oncology, neuro-oncology, radiopharma, and many non-oncological indications. IIG brings with us global operations, so our teams are centered around the world. Technology platform Dynamica for management of imaging data, logistics, and also all of the complicated workflows around dosimetry, radiological review, site management, etc. And of course, we have a great network of partners and radiological readers. So you could see here that, a few facts about us: founded in 2007. We are now about 700 trials in. Our radiological team is recognized through their publication Scientific Impact, top 2% by the World Impact. So today, we are excited to start the session. So I'm sort of going to go fast now, so everybody who wanted to join. And Jared, I think we will invite you to give us an introduction and a bit about Cellectar.
Sure. Thank you. Thank you tremendously for having me here today, Olga, and working with us. So with that, I'll dive in, just to give you a quick background on myself. I'm a physician scientist. I've been here at Cellectar for the last 10 years. I've been working predominantly in radiopharmaceuticals now for approximately 10, 11 years. And as Olga said, I'm the Chief Operating Officer here at Cellectar. What that means in my role is I cover everything from not just supply chain and supply chain management, but also our clinical drug development process and our preclinical programming as well. So it's a pretty broad swath of activities. As we talk about today, we're going to focus in on dosimetry trials and from my perspective, the operational sort of components of it, the challenges both from an operational standpoint, the regulatory standpoint, and patient level sort of considerations as we think about not just dosimetry, but general radiopharmaceutical trials. We will be covering beyond the introduction of Cellectar as a leading radiopharmaceutical company utilizing a novel platform. But also we're going to cover off things such as patient engagement, how to motivate participation in the trials, how to keep them engaged in the trials, site selection, management success to deliver on these trials, because it's not simple. Probably one of the more interesting areas, I would say, is going to be the regulatory expectations. I'm sure as we talk through it, it'll be clear that there is varied regulatory expectation and changes coming potentially, with the whole goal, both from a regulatory standpoint and from an operational standpoint, of us trying to balance these two sort of opposing elements: the scientific rigor of doing good dosimetry and good radiopharmaceutical drug development, but also how do we enhance patient compliance and feasibility of those trials, since we know dosimetry brings certain challenges with it. And then the last piece will really be on the data flow, the systems, timelines, costs associated with these trials, and our sponsors' ability to access that information sort of in real time to make it meaningful. Just by quick way of background, CLERB, as I call it, or Cellectar Biosciences, is a publicly traded company listed on NASDAQ. See the thing there? Obviously, that means that as you look forward to any comments I make about Cellectar are forward-looking statements and should be referred to your investment guides for any additional information about the risks associated with the company. Go ahead. So just by quick high level on the company itself, on high level on the company itself, we are focused on the use of phospholipid ethers or phospholipid drug conjugates, as we call them, PDCs, that utilize a phospholipid ether targeting moiety that allows us to take advantage of the metabolic change and reprogramming that goes on in tumors to allow the uptake of our molecules. Our lead asset is iopofosine I-131 and has been developed in a rare orphan hematologic malignancy known as Waldenstrom's macroglobulinemia, which is a subgroup of the non-Hodgkin's lymphoma branch of hematological malignancies. We did complete a pivotal study a year and a half ago, and we've announced earlier this year, just a few weeks back, top data from that study, 12-month follow-up data from that study, where we achieved statistical significance in that study. It was granted breakthrough designation from the FDA due to that, and prime designation in Europe. We are considering using a conditional market authorization eligibility pathway in Europe, as we also continue to pursue the accelerated approval pathway here in the US, with the expectation that we're launching a phase 3 confirmatory study later this year, with the idea to file for the FDA in the US early next year. In addition to that, we have a number of other programs that leverage that same phospholipid ether moiety and allow us to bring other isotopes to bear. In the case we recently initiated with an Auger-emitting CLR-125 program, phase 1B in triple-negative breast cancer. We've also completed and are ready to launch a pancreatic program with our actinium CLR-225 program targeting pancreatic cancer, as I mentioned. We also have a number of other radiotherapies in development, as well as other programs outside of the radiotherapy space. Next slide. Just to get a quick background, this sort of tells you the story of the company that I just sort of walked through. From a pipeline perspective, you can see, as I mentioned, Waldenstrom's macroglobulinemia is our lead indication. However, we have tested iopofosine across a number of B-cell malignancies, including multiple myeloma and a number of the other non-Hodgkin's lymphomas, such as DLBCL, so forth. We've also tested the drug in high-grade pediatric high-grade glioma because the drug does cross the blood-brain barrier. As I mentioned, we have initiated the triple-negative breast cancer study, as well as are teed up to initiate a pancreatic study either late this year or early next year, and then the earlier stage pipeline. Next slide. So as we think about the challenges or the aspects that relate to dosimetry studies, there are a number of key buckets that I think we sort of laid out earlier, which is, the regulatory expectation. What is really needed here? I don't, not knowing how many have actually attempted this, but obviously when you talk with the FDA, especially as you get deeper into your development programs, what you find is that there's varying requirements, and they're varying based on not just the FDA to EMA, but also inside the FDA, what's the expectation from the solid tumor arena to the hematologic arena. How many patients do you need dosimetry for? What is the correct amount of work that is necessary, and how personalized does this need to get? Even down to educating them on what is the difference between organ level and muscle level sort of calculations. So there's a lot of flux, as what I would say, at the FDA, in particular with regard to what is necessary, what are the requirements, and what level of detail we're going to need for dosimetry studies. And I think, as a group or as an industry, we need to help move them in a certain direction to make these studies more meaningful and standardized. As we think about the other challenges on the operational side of the study itself, these are sort of some of the key components here. The image collection across sites is very complex. As you do this, if you're doing it in 1 or 2 or 3 centers, it's usually pretty easy. But as you move to larger studies, as we did when you go to a global study, the collection of the images is very difficult, because obviously you end up with different centers with different cameras, different specificities, and you end up with different quantifications and image inconsistency. And so, in order to make the data usable and valuable to a company or even to the agencies, we have to get to consistency. We have to get the sites trained to do the same process the same way. If you're working in Florida, you want the same sort of image quality coming out of a Florida institute that you're getting out of a German institute, and you don't want huge variability. Additionally, the harmonization of the quantification is important, and the reconstruction of the PET imaging is critical. What we find is that a lot of times you get this imbalance across sites and changes even with the reader variability, resulting in variability in your data collection and information. So having a centralized, unified process for this becomes very critical. The next is, obviously, this is more of a patient and site challenge, which is, the fact of doing dosimetry requires serial imaging across a number of days. That both has an issue with patients wanting to come back to the clinic repeatedly to be imaged, and it'll allow for both the organ and tumor dosimetry calculations to be conducted. In addition, at the site level, it's very difficult to schedule these repeat imaging, especially as you move to larger studies. In a handful of patients, it's acceptable. When you get to larger, more intensive elements, they tend to become much more restrictive unless they're specialized in doing these types of studies and this type of work. That means that throughout it, there is a huge need for an efficient workflow, not only at the site level, but from sites to central locations. And then, obviously, as I sort of alluded to before, what's really important is that there's a quick turnaround, that from the point of the image being taken, that it can be validated that that image is accurate and is being done correctly. But also, from a sponsor perspective, I want to know and I want to be able to have access to that information as soon as possible to make decisions around further development. And then last, I think correlating, this is a broader issue, again, I think for us as an industry, correlating dosimetry to the clinical outcomes. The key element here is if you look at what's going on, you can see absorbed dose doesn't always align with response rates, if we're looking at objective response rates, or the toxicity we're seeing, nor does it necessarily always align up with durability. And at the end of the day, what is going to be an outcome or an endpoint that we need to be considering and need to be discussing with the FDA and EMA about different outcomes? Do we stick to RECIST and those types of things for solid tumors, or should we be really focusing more on that durability? And how does that play out when you think about a fixed duration therapy versus an indefinite therapy, as is often what we would be compared against in any pivotal or confirmatory trial, where you would be going head-to-head against a drug that's going to be treated repeatedly with radiotherapy. As we know, it's an infrequent dosing and often a fixed duration dosing, which results in a potential for discordant with regard.
Thank you so much, Jared. We will have a Q&A at the end of the webinar, so please feel free to type your questions into the chat because it will be intense on information, and Jared is going through this, through phase 1, through phase 3 himself, so it's an incredible opportunity to ask somebody real open questions. So thank you, Jared, very much. Eric, I'll pass to you.
Very great. Do I have the control of the keyboard, or if not, that's fine.
I'll hand to control. Yes.
Okay, that's fine. Good afternoon. Thanks again for giving us a chance to, Olga, for giving me this chance to talk about the dosimetry. And I'm going to today. My name's Eric Fry. I'm the Chief Science Officer and one of the founders of Rapid. We started about 10 years ago with the dual mission of writing software to make dosimetry calculations easier, and also with doing dosimetry calculations for clinical trials. And today I'm going to talk about the what and why of absorbed dose. I'm really targeting this at people that are kind of new to this area. I see that some of the people on the list aren't, and I apologize for that. But I'll talk about the what and why of absorbed dose, what it is, and why why it might be useful, how we estimate it for radiopharmaceuticals, and a core element of that is the quantitative imaging, and that's something that's a little bit new and challenging for therapeutic radionuclides, so I'll spend a little bit more time talking about that. Next slide, please. So this is kind of a unique time in the history of radiopharmaceuticals. There's a wealth of targets and targeting molecules and chelators. There's a bunch of radionuclides that are now available. This is a partial list of them. And in addition, there's the clinical and commercial success of lutetium-177 dotatate and PSMA-617 that have led a lot of sponsors to bring a lot of new agents to trials. And the combination of all these things, the new radionuclides and all these trials, has raised the bar on imaging. There's a need in these trials for dosimetry, biodistribution quantification, and response assessment. And as Jared said, there's a need to do that consistently across different sites and different scanners. So the next slide, please. So the reasons for estimating absorbed doses, this is to quote Joe Donahue from Sloan Kettering at the PEGS meeting a couple weeks ago, is to prevent disaster. We don't want to kill patients, and so dose is a way to avoid toxicities. And those are usually done using dose limits from external beam. That's quite imperfect, especially for the radionuclides that emit different kinds of particles. But it's at least a way to prevent major disasters from happening and potentially poisoning the whole field. So typically the way that's done is dose estimates from preclinical studies provide a rational starting point for the activity in human studies. And the early phase human dosimetry studies are often, in some sense, verify what was seen in the preclinical studies. So I'm looking at the dose, the fraction of the dose that's deposited per unit administered activity in the different organs, and seeing how that compares to preclinical studies and allowing the future phase 2 and 3 trials. At Rapid, I think not just at Rapid, but there are a number of people that think that the future is potential for radiopharmaceutical therapies is patient-specific dosing based on dose or some things related to dose. And I think one of the things that more and more sponsors are doing in trials is incorporating better dosimetry and maybe for more of the trials. So at least even though the initial dosing may not be based on absorbed dose, that there's information for future trials that might focus on a precision medicine approach. Next slide, please. So let me talk about the foundations of estimating absorbed dose. In nuclear medicine, dose is often used for two things. Sometimes it's used to mean the amount of activity that's administered. So I'll try to consistently use absorbed dose, so I probably won't always do that. Absorbed dose refers to the energy deposited per unit mass of tissue by some radiation. And it's usually measured in modern units in gray, or joules per kilogram. In radiopharmaceutical therapy, the energy is deposited by the decay of radionuclides inside the different organs. And I'll say a few minutes about in a couple of minutes about the different particles that do that. Radionuclides are characterized by a mean energy per decay that's referred to as delta. So if you want to estimate the dose, there are essentially three, and maybe a fourth factor that go into it. The product of three things: the energy per decay. Every time the radionuclide decays, it emits some particles. Some of the particles are absorbed in the organ that it's emitted in. It decays in. Some of them travel to neighboring organs. So the second factor is the fraction of the energy from each decay that's absorbed in some structure of interest in an organ or even a voxel. The third factor is that you need to know the number of decays in that organ or tissue or voxel. And I'll be talking more about that in the next slide, how we estimate the number of decays. The two top factors, the energy per decay and the fraction of the energy deposited in the tissue, are really physics factors that have to do with the radionuclide and the propagation of the radiation inside the body. And both of those are related, especially the middle one, how the energy is transmitted from where it transported from where it's emitted to to neighboring organs, depends on the type of particles that are emitted. Radioactive decay, generally you can think of three classes of particles. The photons. Photons have a low linear energy transfer, so they don't do much damage to the tissues. They have a long range, many centimeters, and as a result, they can be used to image and localize the radionuclide to find out where the decay happened. And then we'll talk about that, how that's done. The second kind of particles are electrons, either Auger electrons or betas. They have higher linear energy transfer. Their range is short, millimeters or even less for Auger electrons. So they're much more useful for a therapeutic application. And finally, alphas. There's a lot of interest in alphas, and that's primarily because they have very high LET. They do a lot of localized damage to cells, multiple strand DNA breaks that are hard to repair. And they have a very short range, measured in the order of tens of microns. In addition, especially for more complicated radionuclides, the alpha emitters in particular, they can have daughters, and some of those daughters can be radioactive. Depending on the half-life of the daughter may redistribute and decay and deposit energy in other organs. Next slide, please. So I want to talk first about how we estimate the number of decays that happen in some object of interest. And if you think about the amount of activity as a function of time, if you're in some organ, the kidneys, for example, after you administer the activity will increase and then decrease as a function of time, and that's known as the time activity curve. Activity is disintegrations per second. So if we want to know the disintegrations per unit time, so if we want to know the total activity inside under that curve, then the total number of decays that occur in that object, then we want to integrate that time activity curve, and the result of that is the time integrated activity. And that can be done using quantitative imaging, quantitative nuclear medicine imaging at various times along that curve. And I think by understanding that, you can understand that you certainly would like to acquire some measurements of the activity at times along the latter portion of the curve. It's a little bit less important to acquire measure the activities at these early time points, because there's less influence on the area under the curve by what happens in this early time points than the later time points. So kind of at a minimum, you'd like to have two or three time points that are distributed, from this peak out, and maybe ideally one at the peak and one at the one before the peak. So maybe four time points closely. Of course, physicists will always like more, but as Jared mentioned, the practical considerations about patients and the cost of doing the imaging and the difficulty for the patients argues for using a smaller number. The shape of the time activity curve depends not only on the biokinetics, but also on the radioactive decay, the half-life of the radionuclide. And so how you need to space those, the acquisitions, the quantitative nuclear medicine imaging depends on both those two things, the decay life, the half-life of the radionuclide, and the biokinetics. So when we want to integrate the time activity curve, we measure the activity at several time points using the quantitative imaging. And then typically the way that the area is estimated is to fit a curve. So usually some combination of exponentials through the activity data, and then analytically integrate those. And then that gives the time activity, the time integrated activity. Next slide, please. So once we have the number of decays, then we can estimate the dose, the amount of energy that's deposited in the tissue. And one way of doing that, one that's accepted by the FDA and has a number of advantages, is the merge style dosimetry. The merge style basically separates the physics from the patient dependent part, the biokinetics. So the time integrated activity gives the total number of decays, and that really talks about the biokinetics and what happens inside the patient in terms of the distribution of the radiopharmaceutical. And now we want to convert that to the dose, and that the way that's done is to multiply the TIA by the S value. And the S values there's an individual S value for each source to target. And what the S value tells you is how much dose is deposited in some target from decays in some source. And the S values can be both, and the sources and targets can be tissues. For example, it could be something like the bladder wall, or it could be the urine. It could be the contents of the colon, and then the target is usually some cells of interest. So, for example, as I said earlier, the bladder wall or the wall of the colon, or the volume of for example, the kidney cortex. The S values are calculated using computational phantoms, and computational phantoms are idealized structures, and the ones that are used for modern S value calculations are quite realistic based on human images. One of the limitations of the MERD dosimetry is these S values are calculated not for a specific patient, but for this generic patient. But especially considering the short range of betas and alphas and those particles, essentially there's not any important difference between the use of those particles, computational phantoms and stylized phantoms, than there are the actual patient, except that the stylized phantoms have different organ masses. And so by correcting for organ masses, you can make the dose calculation essentially as patient specific as using voxel dosimetry. So in order to get as I mentioned, the time integrated activity, we need to do quantitative imaging. And there are two approaches to do that. One is to use some surrogate, and the other is to use direct imaging. A surrogate means that you use a different radionuclide labeled on the targeting molecule. The major advantage of that is that you choose a surrogate that has better imaging properties. So, for example, it could be imaged with PET, or it has properties that are more suitable for a gamma camera, and that typically allows images that have higher resolution, have better quantitative properties, less noise, and so on like that. The downside of the surrogate approach is that the practical problem of they then have to take the surrogate through the approval process before you can give it to patients, and perhaps even more important, the surrogates often have a different half-life, often a shorter half-life, and so if you're trying to do dosimetry for a longer half-life radionuclide, it's harder to get information about what's happening at long time points, depending on what the radionuclide is and the properties of the chelators and so on. You can have a different biodistribution, and especially for things like alpha emitters, one important difference is that you get no information about the daughters. With single photon emission computed tomography imaging, you can sometimes, depending on the radionuclide, you can sometimes get information about both the parent and the daughters. And if you don't have that with a surrogate, then you have to have models to predict where the daughters will go. Quantitative imaging, the direct imaging, is done using, at least in most modern dosimetry protocols, is done using quantitative SPECT imaging. And one of the downsides is that basically clinical SPECT systems are optimized for low energy photons, 140 keV photons. And historically, clinical imaging isn't quantitative. So there's some challenges to do that. So let me talk about that next. Next slide, please. So this is a picture of a SPECT CT system. There's a full diagnostic CT that's combined with a set of rotating gamma cameras. You can then do SPECT and PET on the same, SPECT and CT on the same images, the same imaging system. The CT images are needed for both organ definition and also for some of the compensations, as I'll talk about in a minute, to make the images quantitative. The gamma cameras are rotated around the body after the therapeutic agent is administered, and images are required, projections images are required at a discrete set of projections around the patient. And then the tomographic reconstruction algorithms are used to estimate the 3D activity inside the body. Next slide, please. So if we had an ideal SPECT system, if you have a source inside the body, the photons would be emitted and they would travel through the body without interacting. They'd then be incident on the camera, and the camera would only collect the photons that were perpendicular to the collimator. And if the imaging system was able to do that, then we could reconstruct using direct methods the quantitative distribution of the radionuclide inside the body. Next slide, please. On the other hand, that's not the case. In real SPECT systems, the photons interact. Can you press a return key? Some of the photons are scattered. Press it again. Some of them are absorbed. I'm sorry, go back. And that results in a. Can you go back one slide, please? Yeah. So that results in what's known as attenuation. Some of the photons that are emitted inside the body don't make it and aren't detected, and they give rise to a spatially varying loss of quantitation inside the body. And CT images are essential for correcting for the attenuation. Next slide, please. In addition to some of the photons being absorbed, because the energy resolution of the gamma cameras is finite, when photons scatter, they have a lower energy, but they can still be detected. And that gives rise to tails in the measured distribution of this point source, and that gives rise to a spatially varying loss of quantitative accuracy inside the object. Next slide, please. And finally, real collimators don't just accept photons that are perpendicular to the detector, but they accept photons with a finite angle so that there's a reasonable number of photons detected. For high energy photons that are present in the therapeutic radionuclides, some of the photons can actually penetrate through the walls of the collimator, which are thin pieces of lead. Some of them can scatter and be detected. And the combination of those, the finite acceptance angle gives rise to blurring in the images, and the penetration and scatter in the collimators gives rise to these long tails that reduce contrast and also complicate the quantification. Next, please. And finally, the images with the noise didn't make it through to these slides, but the radioactive decay is a random process, and so that if you take an image of the patient for a fixed amount of time, if you do that multiple times, each time you'll get a slightly different image because of the different number of radioactive decays that occur in the different corresponding voxels. And the result of that is that it makes the images noisy, and that produces inside the reconstructed images is uncertainty, and so that results in uncertainty activity estimates. And the way that works in practice is the fewer counts you have, the more uncertainty you have, and you get fewer counts when you have lower activities. So for radionuclides like the alpha emitters, where the therapeutic effect is so high that you need a low dose, a low administered activity, then you get rise to quite noisy images. Next, please. So just putting this all together, if we in the clinic, the scanners produce a number of set of spec projections. In addition to CT scans, at Rapid we don't use the vendor reconstructions, but we use custom reconstructions that model all the physics and compensate for all those effects in a way that I'll show you in a second. We put those together to do a quantitative reconstruction. That gives a set of 3D image that's a set of slices. It looks like this. This is of a kidney. We want to then quantify the activity inside an organ of interest like the kidney. Then we need a volume of interest, and those are designed usually at Rapid. We use some AI algorithms to get a start, and then manually tune them with the readers. By summing all the activity inside that quantitative spec image, then it gives you the activity inside the object, which then is one point in the time activity curves that I showed you earlier. Next slide, please. So this is, I mentioned earlier that at Rapid we use custom reconstructions to do the quantitative reconstructions, and this just shows basically how all that works. The idea is that we start with a set of measured projections from the scanner, and we start out with initial estimate of the object, and we model all the physics in an iterative process. And in that iterative process, the key thing toward getting a quantitative reconstruction is for this model to accurately model what happens inside the cameras. And so by going through this iterative process of computing projections, comparing them with the measured projections, updating the estimate, and so on, we can arrive at quantitatively reliable projections. And I believe that's all that I have.
Thank you, Eric, very much. I mean, these are incredibly complex concepts, and where we come in as an image analysis group, we try to put end-to-end process to manage radiopharmaceutical imaging trials. What it means is that we are dealing with a number of players in the trial. So we have sites, we have radiology readers, we have dosimetry experts, we have sponsors themselves. And I would like to walk you through that process. And Shreya, after it, will follow up with a live demonstration of Dynamica. Dynamica is our platform that carries that process. So that allows sponsors to have a very consistent, very fast output but also it reduces stress on all of the associated players, including sites, including our partner Rapid. So that just enables a trial to run in a much more standardized manner. So as Eric explained, in a typical implementation of a dosimetry study, you have estimated dose, you will generate a number of documents, you will have site qualifications, that's on the dosimetry side. You would have patient recruitment and monitoring imaging, and then you would be looking at the absorbed dose, which, as Eric explained, will have a specific algorithm. From the full clinical trial imaging point of view, your team is slightly larger. So you normally have a project manager who would be managing distributed partners. So sites will have a coordinator who is ensuring that they can upload images into a centralized location. There will be medical writers, there will be product leads who look at software platforms that is deployed in the trial. You have imaging specialists who are looking at the quality of the images, data managers. And of course, you have two radiology readers and rapid readers who have complementary expertise. So as images flow into Dynamica platform, you have radiologist readers doing first pass, looking at the lesions, looking at the associated tissue. Then the dosimetry can be applied properly to that already outlined information. So we're separating our work between imaging call-up, like image analysis group, and dosimetry call-up, like Rapid. And you could see here a little summary of where we would find ourselves performing, right? So kind of the site qualification, we both participate in the same process, but it's a calibrated effort. So you don't have two questionnaires being sent to the site. So we're working together to make it as simple as possible. We have a system, and now our system, Dynamica, connects to the workflow that enables Rapid to conduct their measurements. You have two sets of specialists. You have board-certified nuclear medicine oncology readers, and you have medical physics, highly experts in dosimetry. So that's very important to actually ensure that within a trial, you have all components over normal imaging clinical trials like audit trail, quality control, response to response, a dashboard. But at the same time, you are capable of integrating a number of experts collaborating to achieve the results. So we always start with a site qualification, right? So we have a number of sites and they have to go through the process to be able to participate in any of the trials. So we are looking at the sites having required licenses and approvals and right roles in places to just conduct such a study. We, of course, looking at the facilities which are associated with nuclear medicine to make sure that these are safe. And sometimes it's a questionnaire, sometimes it's an inspection or collaboration with a sponsor on inspecting the sites. We're looking at equipment. Again, this sometimes comes via a questionnaire, or sometimes you have an inspection. And you're looking at a number of things, from equipment to reading systems, to the age of those reading systems, as they're going to change the system or equipment during the study. And as you know, some of the studies are quite lengthy, especially with a long-term follow-up. So you have to have this information on board. You qualify the staff, and of course that's very important because everyone who's involved in the trial needs to follow the process. They need to know where they're going, what they're doing, where they're uploading images, and so on. We have a quality system and traceability in place, so this is through Dynamica, and we will explain the site, what actually is going to happen to the images. So you have documentation, which explains that. You also have training process, and then you have a final verification. And then, of course, you have a final review and approval for the site to go into the trial. We find that sometimes it's helpful to have one-on-one calls with the site because that's a very complex process. Sometimes it's helpful to have a training center which allows sites to go through the training themselves and then be certified by completing that training. That would include all of the training videos which are relevant to radiology and dosimetry and uploading the data, whatever you need to have in that training portal. That's generally applicable to large phase 3 studies, which will have 50 plus sites, sometimes global, sometimes multilingual. So it's quite challenging not to have something like that in support of the study. Part of the project management is standardization of the study documentation. So as any study would have an imaging charter, certain methodological aspects we would incorporate input both from RAPID and ourselves and radiology physicians. So you would have a very clear guideline on how the training was conducted, how images should be acquired, how they should be quality controlled, and then what kind of reading methodologies we put in place. Communication escalation plan specifies responsibilities and role. Of course, you have data transfer specifications because now you have interactions with the sponsor and CRO and also between ourselves as RAPID and IAG. And there are some supporting documentation. So as the study progresses, that package of documents get populated. So by the end of the study, there is a reliable set of documents to be submitted for any regulatory approvals or further studies. So I suppose this is the workflow which I think originated with our collaboration with Cellectar, and this describes end-to-end workflow for a clinical trial that includes dosimetry. So you could see that in a single integrated data flow, and unique side to this workflow is that every participant can collaborate kind of simultaneously. So we have site image acquisition, and you could see that here we would be acquiring the sites to upload images. Images will be anonymized as the data gets into Dynamica. Once the data gets into Dynamica, there is a second QC check and compliance with the protocol. This is where audit trail starts. You have immediate, as soon as the data lands on the cloud, you automatically start doing audit trail, so you know every step of the way. You then have a radiologist who logs into the system. The data now is on the cloud, so the data is not moving anywhere. And radiologist will specify or delineate target lesions, organs, and so on. It's basically a roadmap for RAPID team to know where to focus their attention. So then you have an export of the whole package of images and roadmap to RAPID, who will be then processing that data, and then results are returning back to Dynamica. So what it allows us to do for the sites to really reduce their workload. I think that the speedy kind of flow for them is just dropping images and with some of the quality checks and then just feedback. Dynamica sits as a data hub and reader tool because it allows us to increase collaboration between the site, the imaging specialist, the readers. The Dosimetry engine is outside of Dynamica, but it has a very tight workflow, which is documented an audit trail between the source of data, the process data, and the final reports. And of course, you also have a real-time visibility on what's going on with imaging in this study. Shreya, I pass to you.
Perfect. Thank you, Olga. Yeah, this is again like a continuation from the previous slide, and it just breaks down the workflow in Dynamica for the Cellectar study. And so before I go on to the demo, I think I'll be just walking everyone on call through the same workflow where we upload images on Dynamica, we QC them, our readers log in, they perform some lesion identification measurements on there. And the interesting part for the Cellectar study was we had to develop this export package, which was then compatible for our partner RAPID to also process for their analysis. So yeah, I think I'll go on to the demo. Quickly share my screen. Yeah. Yeah. Please let me know once it's visible.
We can see it.
Perfect. Yeah. Yeah. Apologies, I'll be off camera due to some network issues, but I hope you'll be able to see my screen. So you're seeing the login page for Dynamica, and as Olga mentioned, it's a cloud-based platform. So as long as you are invited on the platform, you get credentials and you should be able to log in. Now, once the study is configured and it is activated on Dynamica, we invite the site users, and I'll start in by showing how our site users basically upload data into the platform. Okay. So once they log in, they need to select a trial here that they're part of. And it begins with first creating a subject. So they need to create a subject, select a cohort. All of this is configurable in Dynamica. So for example, I want to upload some images in the dosimetry phase. We need to provide some subject IDs that is defined in the platform as well. So as you can see, there's validation to ensure that the site users enter the correct subject ID pattern that is required in the trial. We accept the month and year for the subject. And once we add it, we have the new subject created here. To begin uploading, they need to select this icon, and we take them through a stepwise process for uploading the images. Now we can, by default, like we accept DICOM images in Dynamica, but we can also accept other miscellaneous files and worksheets if required in the trial. So for example, for this study, we require the sites to upload some spec worksheets as well. And so, for example, this is some of the rapid worksheets that the site would upload that has other clinical data for the subject. And so the site users can also upload miscellaneous work files as well. So all they need to do is select the kind of file type. They need to fill in an eDTF, that's an electronic data transmittal form that has information about the month and year for the subject, when was this acquired, and who is uploading this information. They go on next, and they would drag and drop this file directly in here. So we can accept any kind of file formats, all zipped together. All they need to do is just put them in a folder, zip it, and upload it in the platform. And that's about it. So that's how they would basically upload files that are required for the particular subject. Similarly, they would also upload DICOM images as well. And for DICOM images, we also provide a pipeline where there are automated checks. We perform some pseudo-anonymization on the DICOM as well. So kind of like a semi-automated process for the site users to take off the manual load as much as possible. So the DICOM upload also follows a similar process where they need to fill in the eDTF forms, when was it acquired, who is acquiring, and again, drag and drop the DICOM images as well. Yeah, and there we go, upload. And this would take a bit of a time depending on the size of the images. And like I said, our semi-automated DICOM checks are being performed in the background as I speak. All right. So I guess I'll move on and maybe show the next user in this workflow, that is the imaging specialist who comes in. And this is our IAG imaging specialist who performs the basic quality control checks in Dynamica, pretty crucial to ensure that the sites have uploaded the correct images and data required. So let me quickly log in as an imaging specialist. All right, so as soon as the upload is done, it gets directly populated in the imaging specialist's task queue. So there's no wait between the upload and the process for QC. And once it's ready for the imaging specialist, they need to click on the particular subject that they need to perform the analysis on and assign the task to themselves, right? Once you do that, they also go ahead and label these images. So now that's an important feature because we want to be sure that only the labeled correct images go into the next workflow that is for the readers to perform the lesion identification. And so they would log in, open the image viewer. And this is a DICOM viewer where you can see the central view in the center. And we have the images that we have received from the site on the left-hand side. And this is where they would label the images from this drop-down over here. So once the labeling is done, we can go out and you can see some of the labels that have been assigned here. And once they think that, okay, these images are ready to be sent to the reader, they click on this task, the reading task, and this then goes ahead to the reader's queue. So it's, as you can see, it's pretty quick. There's no wait in between as I'm going through one step after another. I think it's also interesting to show that we also support queries, sending queries to the sites as well. So in case if certain images are not as per required, they can also send certain queries. For example, if there's some missing information, some missing image data, we can send that to Dynamica as well to the site users. So I can just put in some text for the site users to respond to and confirm. And so even we have that query management happening within Dynamica. So all of this gets audit trailed as well if you want to go back to it. So yeah, that's basically the imaging specialist workflow in Dynamica. I think I'll move on to the next user in this workflow, that is our readers coming in.
And of course, as Shreya opening that, all of this is happening simultaneously at all times. So the readers, the sites, the imaging specialists. So per day, we see thousands and thousands of scans loading, reading, receipt, and so on.
Yes, that's right. So as you can see, this is the readers list. And this was a reading I just triggered in the previous step. And so the reader would have to ideally click on the reading task, and they should open up the same image viewer. Now, the only difference here is they will see only the labeled images that was QC'd in the previous step. We have the same central viewer, and they can begin to start marking the lesions. So on the right-hand side, we provided a very basic electronic case reporting form where they need to provide a basic image quality assessment. So if they think if the image quality is optimal, and to begin drawing the ROIs, they would click on this particular card, and this would launch the segmentation tool. So we've kind of integrated the segmentation process within the same viewer. As you can see, there's no additional installations or any other kind of viewers that is required to perform these processes. So once we call in the segmentation process, we provide some basic segmentation tools like a brush, and they would ideally start drawing on the image here. And that would get computed, like the volume of the segmentation that I'm drawing would get computed. And as you can see, it's been captured automatically in the case reporting form as well. I just hover over the segmentation, and we have the other statistics regarding the voxel count, the volume, the min-max values as well for the segmentation I just performed. And so once the reader has identified the required lesions that they want to assess, we would draw the lesions and they would lock the ROIs. So this ensures that all of this data is getting saved, it's going into the backend. And also what's happening is once they submit it, the packaging begins. That is the packaging for our partner Rapid team to export from Dynamica as well. So this is basically how the reader would interact with Dynamica in the same platform here. And as I mentioned, I can quickly also show you how the exports can be accessed. So for example, I'll log in as this other user role. We call this the external user role. And this is where they would ideally log in and export out all this reading information, the miscellaneous files for further analysis. So let me just log in as this user. And so as an external user, they would see the list of subjects, from which site they're from, what cohort, what was the last uploaded visit, and when was it uploaded. So if I need to search through some subjects, I can search. And as you can see, we have our reading results that was submitted by our readers. That will be in a DICOM format, and the segmentation files, and the uploaded miscellaneous files by the site as well here. So this will be available to download from Dynamica. And the further analysis for dosimetry can be conducted using these files. And once we have the Rapid team done with the analysis, they have the reports ready, they can log back into the platform and also upload results on the same subject. So as you can see, we've provided the upload report within a particular subject for a particular visit to ensure that it all gets tracked accurately in the platform. So they can upload a report here. Again, we'll accept different kinds of file formats. And upload one here. And there you go. So we have a new report upload on the platform. And I think finally, all of this process can also be seen on the dashboard as well as a sponsor. So I'll quickly show in the sponsor dashboards, which would give in a nice overview of how the subject has progressed through these different steps I just showed you. So let me log in.
Because the sponsor is their own user, because some of the sponsors allow to see certain things, some have different requirements. So for every sponsor access, we configure that separately, so to make sure that it's in compliance with any rules that sponsor has internally.
So what you see here is this dashboard that even our internal project managers can view, and the same dashboard that our sponsors view as well. So this gives a little breakdown of at what stage of the process the subject is. So for example, I can see what's the QC status, when was it completed, was a reading generated, when was it completed. Like Olga mentioned, we can also provide access to completed readings. So if a sponsor wants to see the lesions that were identified in the viewer, we can also provide that access where they can click on the completed reading and also see the ECRF that was completed by the readers as well. So all of this flexibility can be toggled on if required for the sponsors. And I think finally, compliance being such a hot topic across trials, we also have our audit trails and electronic signatures in place to ensure that trials are compliant at the end of the day, and we don't miss out on that aspect. So yeah, I believe that's about it. Yeah, I just wanted to also maybe show the audit trails. So exactly, yeah. So we can see all of the actions I performed right in this demo. We can see who performed when, what was the particular action type. All of that can be drilled down in the audit trails here as well.
That's superb, Shreyas. Thank you. And thank you for finishing right on the dot. So what we have prepared for you is a short list, is a checklist of everything you could possibly need to run your next study. As we don't have much time to go over it, I really encourage you to reach out. So you can reach out to any of us. You have this contact email address, which please feel free to use, or send me a note. And we would be very happy to follow up and give you answers. I do see some questions on the chat, but I appreciate of people's time, so I'm probably not going to hold this for Q&A. So we will respond individually. So thank you very, very much for attending and being with us for the full hour. I could see very familiar faces. So look forward to seeing you again and working together. And thank you so much to the speakers, Jared and Eric. Thank you for the time and collaboration.
Thank you.
Thank you very much. Thank
It's a
you,
pleasure.
everyone. Bye-bye. Bye-bye.